We will then move on with the program and perhaps take a step to the side and go back to the systems that the fish AI prototype and device hopefully is going to replace or substitute, which is the live animal feeding trials that have been introduced briefly during the earlier talks. And of course, many would say that the usefulness of an in vitro model will very much depend on the in vivo, in vitro correlation. So what we will hear here now in detail is a presentation of one of the feeding trials that were conducted in parallel with all the work done on cells, with, as has been mentioned before, exactly the same type of diets. So making this in vivo, in vitro comparison efficient. So the next speaker will be Guro Løkka from NMBU. Guro is a researcher here at the Faculty of Veterinary Medicine at NMBU, belonging to the Nutrition and Health Research Unit. She's been working here at the vet school since 2009. Her background is a PhD on Atlantic salmon gastrointestinal anatomy and immunology. Her research is mainly focused on mucosal surfaces of cultivated fish species and the links between nutrition, immunology, and gut health and function. And the recent years, also aligning with the fish AI activities, her focus has been on development of in vitro models to reduce the number of live animals used for research purposes. So please, Guro, the floor is yours.
Thank you, Trond. I have to turn this on. Okay, now we're there. Thank you, Trond. Welcome back, everyone. So I will continue on the topic research strategies for evaluating fish feed ingredients and fish feeding trials as reference for in vitro studies. So I will start with some introductory slides, going back to the background for the project that Fulvio talked about this morning. So I will start to remind you how the feed for carnivorous fish has changed dramatically over the last 30 years. So this slide shows the feed composition for Norwegian Atlantic farm salmon from 1990 to 2020. But the picture will be exactly the same for rainbow trout and also for other carnivorous fish species, including the marine species such as sea bass and sea bream. And the reason why I use Atlantic salmon as an example is the enormous numbers we have for this industry in Norway. So, as you know, the diet has changed from a completely blue marine-based diet until an increasingly more and more green plant-based diet. And today's salmon diet consists of about 75% vegetable ingredients. 20% of this is soy protein concentrate, 18% rapeseed oil, and 15% wheat. And the reason for this huge shift is, of course, the limitation of the marine resources. So there's a global competition for these resources, for human consumption, for pet food, and for other production animals. So the resources are limited, and also then the prices increase. And the thin yellow slice that you see here is what's called other feed ingredients. It's now a very small fraction, which can be ingredients such as microalgae, single cell protein, and insects. So these new alternative and sustainable feed ingredients, they are more and more in focus. As already mentioned, we have some examples here: insect meal, single cell protein, microalgae, macroalgae. We have plankton, grassland protein even, and we have krill, and also these metapelagic species. And these are all examples of new and sustainable fish feed ingredients that can be produced or harvested where there's less competition for the resources. So sustainability is a key word here. When looking for these new feed ingredients. In Norway, in fact, the Norwegian government, they stated in 2021 that all feed for the aquaculture industry should come from sustainable sources by 2030. This has already been pushed to 2034. And several reports have been published, and this goal is defined now as a common mission for society, where research is a very important contributor. So what does this mean? Is it realistic if we look at the Norwegian salmon industry? If all aquaculture feed should come from sustainable sources, what does this actually mean? So today, Norway produces 1.5 million tons of salmon, meaning that we need 1.8 million tons of feed. Of this, about 500,000 tons is protein and 500,000 tons is fat. And if we are including 19% soy protein concentrate, we would need 600 to 700 tons of soybeans. And with the ambition in Norway to by 2050 produce 5 million tons of salmon, we would need 6 million tons of feed, meaning 2 million tons of protein and 2 million tons of fat. So as you see, these are large, huge quanta. So when we are considering new raw materials, the availability of this product or the upscaling in the production is a really important factor to consider. And the need for research and innovation is large. And if we look at the future scenario for Norwegian salmon feed, we see that by 2050, most of the feed is predicted to be contained of these other feed ingredients, the green part of the bar here, while the inclusion of soy is decreasing. And probably this, it will not be like one new super ingredient, as the soy has been up until now, but rather a combination of different other feed ingredients. So what are the steps for evaluating a new feed ingredient? I will go through the steps in a paper by Glenn Cross from 2020, where this process was described to include 7 steps. And you will see here is some overlap with what Ramon talked about earlier this morning that they are doing now in the industry. So the first step is to characterize the ingredient. So you have the chemical composition, variability in composition, source and species, and also availability I put under here, as I already talked about. So the picture here shows a catch from a mesopelagic species from another project as a fee harvest. And as you see, it's a large variation in the catch. And also you can imagine you will not get the same every time. So you need to do this characterization every time. Then you have the palatability. How the feed tastes to the fish is, of course, very important, as illustrated by this example, where more than 80% of the weight gain for the fish was explained by the feed intake. So if you have an ingredient that increases the palatability, so the feed intake will increase, then this ingredient will have added value. And in contrast, if you have an ingredient that reduces the palatability, it will be of less value. Then the next step is the digestibility, which Ramon talked a lot about earlier. So this is the measurement of the proportion of energy and nutrients which an animal can obtain from a particular ingredient through its digestive and absorptive processes. And you measure then the relative disappearance between the nutrients in the feed and in the feces. The figure here shows the energy flow through the animal. So you have the energy in the feed, then you lose some to the feces, and you have the digestible energy. Then you will lose even more to the urine and also to heat maintenance, and you will end up with a net energy. So here we come to the next step. Also, Ramon talked quite a lot about what can actually be utilized from the nutrient by the animal. So what can be retained. And then you have effects on health and welfare. So an ingredient can have either positive or negative effects. The picture here shows inclusion of soybean meal, giving the soybean meal-induced enteritis in the distal intestine of Atlantic salmon. So when you have the unrefined soybean meal, the antinutrients cause this reaction in the intestine. So now they have to take out the antinutrients and only feed soy protein concentrate. Next step is processing effects. So an ingredient can, of course, influence the physical properties of the feed. The fish feed are extruded, so the ingredient need to tolerate this process, and also factors such as the storage and flow needs to be assessed. And then finally, you have the product quality. So the ingredients in the feed will can affect the smell, the taste, and the color of the fillet. And you also have the sensory qualities like the firmness and gaping of the fillet. And you also have these the sustainability measures. If you can label that your end product comes from sustainable ingredients, this might have added value for the consumer. So some of these steps are not easy to model in vitro, such as the palatability and the product quality. So what we have focused on in the Fish AI project and has already been presented by my colleagues earlier is step 3, digestibility and effects on health. That will be the local gut health. So when we are developing in vitro models to evaluate new fish feed ingredients, we need to validate it against in vivo results. If we cannot, if the results from the in vitro model is not comparable to the in vivo results, then the model has no value. So we needed to run in vivo trials, already mentioned, and the aims of these studies was to test contrasting commercially relevant diets for rainbow trout that we worked on in this project to provide in vivo reference for in vivo testing of the same feeds. And in the project, we ran 3 feeding trials. So the first trial was with the rainbow trout of about 1 kilo in salt water. There we only had 2 contrasting diets, fish meal and 1 diet with soybean meal. As I mentioned, this soybean meal is presumed to induce intestinal inflammation in the distal intestine. And then we ran a second feeding trial with smaller rainbow trout in freshwater. We had, again, a diet containing fish meal, high level of fish meal, and then one with soybean meal. And we also had 5 diets with challenging ingredients, guar and feather, at different levels or in combinations. So this is the feeding trial that Amir talked about, where we compared the in vivo results to the in vitro digestion. And based on the second feeding trial, we ran a third trial with a bit larger fish, also in freshwater. And we chose 3 contrasting diets from the second feeding trial. So we had fish meal, soybean meal, and this feather meal. So we had the high feather diet with a lower digestibility. So these 3 diets we had already tested. So we knew from the previous trial, we saw that they were contrasting. And then we added a new product, a single cell protein meal in either 10% or 20% inclusion. So this third trial is the one I will present results from now. And that is what will be presented by Fulvio later that we compared against the in vitro results concerning the health. So the diets were formulated by Skretting. So we had the fish meal diet, presumed an optimal diet. Soybean meal, presumed to induce the intestinal inflammation. So this was our bad health diet. And then we had the feather meal diet with presumed lower digestibility. And then we had these two diets with the single cell protein at 10 or 20% inclusions. And this, it was a product selected by Skretting called Coran Pro, but with unknown characteristics. So we had the 3 reference diets and this more unknown ingredient. And as you see, there are also variations here in other of the components, such as wheat gluten, and soy protein concentrates. So the diets were formulated to be isoenergetic and isonitrogenous. So these were balancing the diets that was necessary to do. And also, the focus here was not to have, for a regular feeding trial, this would not be the best, but here the point was to have these contrasting diets that we could compare against for the later in vitro results. So then we have the experimental design. We have the rainbow trout of 160 grams. We had triplicate tanks. And we fed the fish the 5 different diets for 9 weeks. And we obtained samples for growth performance, digestibility, blood biochemistry, organ indices, histology, and gene expression. So if we first look at the fish performance and growth, we have the final weight of the fish, the specific growth rate, the feed intake, and the feed conversion ratio. So the fish performed well from all test diets. And we see that the fish fed the single cell protein diets, they performed equal to the fish fed the fish meal diet. Also, the fish fed the soybean meal diet, they performed well, equal to the fish meal fed fish, while the fish fed the feather meal, they actually had an increased feed intake, and they also had a higher final growth. But looking at the feed conversion ratio, there were no significant differences between the fish fed the feather meal and the fish fed the fish meal. So these results could be explained by a lower digestibility of the feather meal diet, where the fish are compensating for this reduced digestibility by increasing their feed intake and resulting also in an increased growth. So let's look at the protein digestibility numbers. For fish meal, soybean meal, and the single cell protein diets, protein digestibilities were above 90%. So it's the sum of amino acids I present here. While for the feather meal diet, as assumed, the protein digestibility was suboptimal, about 6% lower than for the fish meal diet. Also, the single cell protein diets, they had significantly lower protein digestibilities compared to the fish meal diet. And this effect was dose dependent. So looking at the limiting amino acids, cysteine, histidine, lysine, and threonine, we see the same trend. For the feather meal, the fish fed the feather meal diet, the digestibility was significantly lower. While for the single cell proteins, it was only the 20% diet that was significantly different from the fish meal. So this underlines the dose dependency of this ingredient. Then we looked at the blood plasma biochemistry. So we have free fatty acids, glucose, and triacylglycerides. There was no significant differences between the diets, and all values were in normal range. But for the cholesterol, we saw a significantly lower level in the fish fed the soybean meal. This was expected since the cholesterol level is known to decrease with increased soy, and also the soybean meal itself doesn't contain cholesterol to the same degree as fish meal. And then enzymes related to liver function. We saw increased increased levels for the single-cell protein diets, but these differences were not significant due to large variation in this groups. So it's hard to conclude anything from these results. Then we looked at the organ indices, meaning the organ weight related to the overall weight of the fish. So we have the somatic index of the stomach, the pyloric intestine, mid-intestine, distal intestine, and liver. And we saw a reduced organ index for the fish fed the soybean meal in the distal intestine, and also a similar trend in the mid-intestine. And this is the first sign we see when we see this intestinal inflammation in the distal intestine, that the weight of the distal intestine is significantly reduced. So then the next step was to look at the histology. So I have already mentioned this soybean meal-induced intestinal inflammation several times, so I'll just show you these signs that we are looking for. So the picture to the left shows the normal mucosa, while the picture to the right shows severe inflammation. And the signs that we score when we do this evaluation is increased cellularity of the submucosa. Then we have also increased cellularity of the lamina propria, so we have a widening of the folds, a reduction in the fold height, and finally a loss of enterocyte vacuolization. So then we scored histological sections from the distal intestine of 18 fish per group for these 4 characteristics. So we have the submucosa cellularity, the lamina propria cellularity, mucosal fold height, and supranuclear vacuolization of the enterocytes. So the scoring system is from normal, indicated by blue, and then with mild, moderate, marked, or severe changes. So the more red the bars, the worse the inflammatory state. And as you see, the fish fed the soybean meal diet had a significant and clear inflammation in the distal intestine. But also note that the fish fed the single-cell protein diets, they showed some stimulation, immune stimulation or mild inflammation for all these 4 characteristics. Finally, we looked at gene expression also from distal intestine tissue. So then we looked at a gene with function in water uptake, lipid uptake, and brush border for brush border function. And we saw for all these genes, it was significantly reduced in the fish fed the soybean meal. And this indicates that there is a loss of these normal functions in the fish, of course, due to this inflammation. We also looked at some immune genes. So we looked at, we had interleukin 1 beta, interleukin 8, and TNF alpha, where we saw either an upregulation or a tendency of a higher expression, which can correspond to what I showed from the histology, that you have a mild immune response from this single-cell protein ingredient. So to summarize the in vivo trial, the single-cell protein diets performed equal to fish meal concerning growth, but slightly lower protein digestibilities. So this product may partially replace fish meal in fish diets, but probably not more than 20%, maybe not even 20%. And we saw this mild immune stimulation in the distal intestine, but whether this is positive, like an alertness of the immune system, or negative, like a mild inflammation, it's not possible for us to conclude from these results. And we also confirmed this intestinal inflammation from the soybean meal diet, and we confirmed the suboptimal digestibility for the feather meal diet. So the experimental setup with the 3 contrasting reference diets was successful for testing new feed ingredients. That was the single-cell protein in this study. And also we obtained results that provided an in vivo reference for testing the predictive value, the predictive ability of these feeds in vitro. However, when we are trying to rank the feeds, what is the best, what is the worst feed, this will differ with the biomarker we choose. So if we first rank them by digestibility, the fish meal and also the soybean meal showed the best digestibility, followed by the single-cell protein diets in dose-dependent manner, and then finally the feather meal had the lowest digestibility. If we rank them by growth, then the feather meal performed the best, followed by the single-cell protein 10, and then fish meal and single-cell protein 20, and then the soybean meal on the bottom. If we rank them by the feed conversion ratio, then the fish meal and the single-cell protein 20 will be on top. Single-cell protein 10 follows, and then feather meal, and finally soybean meal last. And if we rank them according to the health parameters, so this is both the histology, the gene expression, and the organ indices, we would rank fish meal and feather meal on top, followed by the single-cell protein diets in a dose dependent. So increase the dose will push you down the list, and then the soybean meal performed last. So the conclusion is that the quality of a feed ingredient is a sum of many characteristics. And to rank the feed from an in vivo trial is not like a straightforward. It depends on which biomarker you look at. Yes. So this study has been submitted to aquaculture reports, so we hope it will be out soon. And I will thank my colleagues in the consortium. Yeah. And be happy to take any questions, of course.
Thank you, Guro. Any questions from the audience? I see a hand up there. Please introduce yourself. And yeah, we have a microphone. See if it's working. If you can, please wait with your question until you have the microphone. Then the people joining us online will also be able to hear you.
Where was the question?
Question up there.
Yeah.
First of all, thank you. Like, your presentation is like really nice presentation. My first question is that what is the like main reason why soybean meal is causing like inflammation of distal intestine, and is it causing it in another species as well, just like large animal, cattle, or buffalo, or these kinds of animals? And second question is that like is there any other sustainable ingredient that can replace soybean meal except the single cell and feeder meal that has a potential to replace soybean meal in future?
Yeah.
Because I have seen on the graph, and like it's looking like that up to great extent new ingredients will replace the currently used ingredients. So these are my two questions.
Yeah. Okay, so the first one, we have the expert on the soybean meal-induced enteritis, Åshild Krogdal, with us here today. So you can talk with her during the lunch break for all your questions. But the short answer is that it's the anti-nutritional factors and especially the saponins that are causing this reaction. And in salmonids, this reaction is very bad. So the Atlantic salmon, but also the rainbow trout, while other fish species, such as the cod, they don't react. Is that correct? Yes. When it comes to other animals, I cannot answer. So maybe Åshild can have an answer for that, or some of the other colleagues. I don't know. I'm very focused on fish. I'm sorry. And then your second question was if any ingredient can replace. Yeah, so this is sort of the big question, because I think that probably no, you have to have a combination, but the industry can also maybe answer better which are the most promising candidates right now. But I think the different single cell protein products and also insect meal are already used, and also we have Margaret Øverland with the Foods of Norway. They have been working a lot with upscaling of this production, because that is sort of when you find an ingredient, to upscale the production to actually get these really large quanta that you need for the industry. That is a really important factor here. So yeah, and maybe there are also some ingredients that we haven't found yet.
Thank you. Yeah, we are running slightly over time, but it's okay. Please, we take another question.
Hello, thank you for your nice presentation. I'm Vahid from Sweden, University of Borås. My question is that, as you said, that's for strategies for ingredient evaluation, and you had 2 levels of inclusion, 10% and 20% of fish meal inclusion. And did you evaluate it before, how it's close to the fish meal? For example, in terms of amino acid profile, your ingredient or single cell protein, it's fish meal. Because at the end you said that's not more than 20% can be included.
Yeah, that was based on this, that you see this immune stimulation. So that's like sort of also that you see a slightly lower digestibility with the increasing level, and you also see an increase in the immune response. So then you would think that if you go more than 20, you will probably both decrease the digestibility and also maybe get to and even if it's a slight inflammation, this can get worse. But it was just the first trial. So.
Okay, thank you.